I want you to focus on the summary slides for the pathways. There's a lot of information on those summary slides, and I'll show you one example of one. And then including the rate limiting enzymes, those are always going to be very important. That's a low-hanging topic question that's typically on the exam. And also, we talked about a lot of intermediate activation steps for the pathways. The oxidation of pyruvate, the activation of fatty acids. So those are also very important. And then if we talked about any diseases or conditions in any significant detail, please pay attention to those and know those. Also, any drugs. Drugs are, you guys are going to learn 1,000 drugs while you're here. So when you get started this semester to kind of distribute it, I guess, a little bit. So anything with drugs, we talked about several different drugs that are used. Know that. And also, I forgot to put it on this slide, but there's a table, and I highlighted it as something you guys should focus on. Please do. For example, the glycogen storage diseases. You may see some questions with that information that's made. Any questions about how to focus your studies? One thing I don't want you to focus your studies on is in the second lecture when we talked about the different roles of the lipid phospholipids, there's a table for that, like cardiolipin and all that stuff. Don't focus on that. All right. Questions? In the second lecture, we talked about 5 different most common membrane phospholipids. Cardiolipin was one of the examples. Don't focus on that table. All right. Any questions? Yes.
So you mean like it's like a bunch of slides about like individually what they do, so you don't focus on those slides?
Right. Don't focus on the slides or the summary table. All right. Any other questions?
Which lecture was that, sir?
The second.
I'm trying to
No, it's fine.
I'm just trying to remember.
It's fine. Y'all have a lot going on. So.
Gosh.
Any other questions? All right. So we talked about a lot of different pathways in this block. That's hallmark for biochemistry, metabolic pathway. These are all the ones that we talked about in this block. And so with that being said, we're going to start with some summary questions. One person, please tell me what the most important products of the hexomonophosphate. Yes. And beautiful. NADPH. All right, great job. Thank you. Any questions about that? No? Okay. All right. This is one of those intermediate steps that I was talking about. So the oxidation of pyruvate is very important. Also remember where these steps take place in the cell. Where does glycolysis take place? The cytosol, correct. So in order for the TCA cycle to begin, that pyruvate must be transported into the mitochondria, okay? And so what happens is, when it does that, it interacts with a coenzyme A in the presence of an enzyme that we're going to talk about on the next slide to produce acetyl-CoA. Acetyl-CoA is the intermediate for the first step of the TCA cycle. So, very important, the oxidation of pyruvate. Then you have acetyl-CoA, and what molecule makes citrate? Oxaloacetate, right? So that is the first step of, or the preliminary step for the TCA cycle. And then you produce citrate, which is how this pathway gets its name. as we go through the turn of the TCA cycle, the most important components, this is the summary slide that I was talking about. The most important components are the rate limiting enzymes, and not actually which step it occurs, but what the rate limiting enzyme is, which is isocitric dehydrogenase. Whenever you see a dehydrogenase enzyme, there's going to be an NADH produced. Okay, so that is a clue that you're going to get your first NADH from the TCA cycle. Remember, the TCA cycle produces NADH, FADH, and GTP, which is converted to ATP, which NADH and FADH will be later oxidized in order to produce energy. So we get 3 different NADHs throughout the TCA cycle. Alpha-ketoglutarate is very important because, and we'll talk about this when we talk about the metabolism of proteins. And we'll talk about how that's involved in transamination reactions. So I just want you to be mindful of the interplay or the connectivity of all these different intermediates. These intermediates, you've seen several of them in block 2 that appear in different pathways. Alpha-ketoglutarate, fumarate. Remember we talked about malate as being a transport system for electrons to get into the TCA cycle. But the take-home message for this slide is for one turn, and that is with one pyruvate molecule, we will get 3 NADH, 1 FADH2, and 1 GTP. Are there any questions? Yes. The GTP will be converted to ATP in another part of the cell, so it's equivalent to ATP. Any other questions? So that NADH and FADH2 will be transported to the mitochondria to produce ATP. So there are 4 complexes. 4 complexes in the TCA cycle, in the ETC cycle. And these complexes, 3 of them are responsible for transporting hydrogen ions into the intermembrane space. Those are complexes 1, 3, and 4. So what happens in the ETC cycle, NADH will donate its hydrogen and electrons to the first complex, which will the hydrogens will be pumped into the intermembrane space. The electrons from NADH will then be donated to the electron transporter CoQ. CoQ will then donate those electrons from complex 1 to complex 2. Or I'm sorry, FADH acts only at complex 2. NADH acts only at complex 1. So FADH will donate its electrons to complex 2. CoQ will take the electrons from complex 1 and complex 2 and donate those to complex 3. Again, more hydrogen protons will be pumped into the intermembrane space. This process is building the gradient that will be the driving force for ATP production. So complex 3 has all of the electrons from 1 and 2, and it will donate its electrons to the cytochrome C, which will then donate all of those electrons to complex 4. Complex 4 will become supercharged by all those electrons, further facilitating protons to be pumped into the intermembrane space. At this complex of very importance is that this is the complex where oxygen becomes the final acceptor of electrons, producing a water molecule. Because of this proton gradient that serves as the driving force for all the protons to now go down complex 5, which is ATP synthase, to produce ATP. Now, steps, complexes 1 through 4 represent chemiosmosis, which is building that proton gradient, which is the driving force. Complex 5 is oxidative phosphorylation. The whole process is oxidative phosphorylation. So complexes 1 through 4, chemiosmosis, together all of them are oxidative phosphorylation. And what that means is that the coenzymes are being oxidized to eventually phosphorylate ADP into ATP. Are there any questions about the electron transport chain? Yes.
Where are the hydrogens in complex 3?
They're the hydrogens in complex 3, so FADH2 cannot donate hydrogens because it's not a transmembrane complex. So there's going to be hydrogen ions floating around in the mitochondrial matrix. So that's what's going to come from FADH2 and other molecules.
I thought the hydrogens from FADH were used in complex 4 to make the H2O.
Yes, that's true. So
They just kind of split.
this picture doesn't show it, but there's also other hydrogen ions from our protons from other interactions, reactions occurring. So it's coming from the NADH, FADH2, and other substrates, I guess you could say. And so that's how there's so many that enters the intramembrane space to create the driving force for ATP synthesis. Any other questions? Yes.
So the slide said that
Complex
complex 5 was chemiosmosis.
5 was chemiosmosis. So chemiosmosis is the building up of the proton gradient, and that's done by complexes 1 through 4. So let me hold on one second. Let me pull up that lecture. Let me tell you guys. Oh, yes, I'm sorry. I said that. My apologies, I said that. So, what happens between complexes 1 and 4 is the building of that proton gradient. And then when the protons go down complex 5, that's actually chemiosmosis. Together, all the complexes are oxidative phosphorylation. Does that make sense? My apologies. Any other questions about the electron transport chain? Yes.
I just want to make sure I kind of understand the big picture. So we're building, so 1 through 4 is building the proton gradient between the outer and inner membranes.
Yep.
Because the oxidative phosphorylation process requires a lot of energy. You have to get big concentration gradient that way, shove all that energy through complex 5. That's where you get your big energy.
Correct. And the whole process is oxidative phosphorylation. So complexes 1 through 4 is actually the electron transport chain. Complex 5 is chemiosmosis. All together, they are oxidative phosphorylation. That's what I meant to say. you for that. Any other questions? Yes.
And then similar to the engines, is the phosphate ones, are those just like free flows?
Yeah. From other reactions going on. Yeah. Any other questions? So we talked about a number of lipid pathways, either to build lipids that's lipogenesis or to break those down for energy sources. Of key importance is all of those pathways converge at acetyl-CoA. So acetyl-CoA is either substrate, intermediate, or the final product of the pathways that are involved in lipid metabolism. So we talked about lipolysis. We know that is the breakdown of triglycerides. So what happens is that glycerol backbone here will become intermediates of glycolysis or gluconeogenesis in the form of dihydroxyacetone or glycerol dihydrophosphate. And we know the fatty acids will be further broken down via beta oxidation to produce acetyl-CoA, which will then enter the TCA cycle to produce energy. All right, any questions about that?
Is there a name for the glycerol? I'm sorry. Is there a name for glycerol that gets transferred over to the glucose 3-phosphate or DHAP? Is there a name for that process?
Is there
Like
a
that-
pathway?
Yeah, is there a name for it?
I don't, if there is, I don't.
Okay. Enough for this. Got it.
So beta oxidation, the breakdown of those 3 fatty acids from that triglyceride into acetyl-CoA molecule. So in order for beta oxidation to occur, the fatty acid must enter into the mitochondrial matrix. And how it does this is that carnitine shuttle system. So the carnitine shuttle system will take that fatty acid that has already been activated by the CoA, remember that activation step. So fatty acyl-CoA will, that process will remove the CoA and add a carnitine moiety, which will then allow the fatty acyl group to enter the mitochondrial matrix through a carnitine transport shuttle system. And so that is catalyzed by carnitine acyltransferase 1. This is the rate limiting step of this system. So what happens when that fatty acylcarnitine group gets into the matrix, the reverse reaction will be catalyzed by carnitine acyltransferase 2, removing that carnitine group for further use, adding back the CoA. So this fatty acyl-CoA now in the matrix can begin beta oxidation, which is removing 2 carbon molecules at a time to produce acetyl-CoA. Any questions about this shuttle system? Yes.
Sorry, so Cat1 and Cat2, are those different rate limiting enzymes, or is that just saying that's the first time it's used in this process? Like, is there a core distinction to make?
Yes, because Cat1 is the rate limiting enzyme. Cat2 is just a similar enzyme that's going on. So make sure that was a good question. Make sure you see that again. Cat1 is the rate limiting enzyme. Cat2 is not the rate limiting enzyme.
Is it rate limiting? Is it based off the amount?
Yeah, based off the amount and the driving force. So if we need to use our fatty acids, that means we've exhausted all of our glycogen and all that stuff. So that rate limiting enzyme means it's like the bottleneck. So it's going to determine how fast the reaction goes. So if we need that energy, it's going to be fast. Does that make sense? So there's already going to be a production of that protein. It's going to be upregulated. Yes.
So is it inhibited by like ATP levels like the other
That's a great question. If we have enough, it could be, yes. Yeah. But remember, when we're using this source of energy, so carbs are our first primary energy source because they're easier to metabolize. This is the secondary, and then amino acids are the third energy source that our body uses, and of course, going by the increasing difficulty to break those down. Yes.
So as far as for products for this, I was just assuming it's palmitic acid, like you said that the slide shows, so the eight OAs, whatever, and then the 7 ads and the 7 FADs.
So great question. And so you can. So remember, we're breaking down fatty acids here. So you can assume that it's a 16 carbon fatty acid, which is palmitic acid. Typically, that is the maximum number of carbons that is built during lipogenesis, and we're going to talk about that. Great question. So palmitic acid is a 16 carbon fatty acid that is the most we can make during the standard lipid metabolic pathways that we're talking about. So, any other questions about the carnitine channel system? All right, so if we have any deficiencies in proteins or enzymes involved in beta oxidation, we will have a beta oxidation disorder, and that can be myopathic CAT2 deficiency. And so what that means is that the fatty acyl carnitine group has entered the matrix, but we can't remove that carnitine group and reattach the CoA. So we can't do this step right here. So what that's gonna lead to is increased triglycerides in the muscle because we can't break them down. That's gonna cause our muscles to be weak because we're not getting any energy. We can't produce energy, hypotonia. And then it's also gonna cause our muscles to break down, and then we'll see myoglobin, which is a protein in our muscles that will break down and it will spill over into the urine. So we would treat this by having the patient have a diet higher in carbohydrates, so they will use that as their main source of energy. All right. So the systemic primary carnitine deficiency disorder is when there's the shuttle system is deficient. So what happens is you would treat that with the over-the-counter L-carnitine. And so if you don't have this carnitine shuttle system, that means we can't transport the fatty acids into the matrix. So again, you're going to see symptoms as a result of that. Muscle weakness or hypotonia. You're also going to see hypoketotic hypoglycemia. So we're already hypoglycemic because we don't have any glycogen, we don't have any glucose. So we're using fat now for our energy. So, but then typically we would see a rise in ketone bodies because beta oxidation will produce ketone bodies. And so because we're not able to oxidize our fatty acids through beta oxidation because we can't get them into the matrix, you're going to have a hypoketotic state. So you will have hypoketotic hypoglycemia. Any questions? All right, and to your question earlier, so for fatty acid synthesis, great question. That process can make up to 16 carbon length fatty acids, which is palmitic acid. And this is a process called lipogenesis. this occurs in the cytosol. Where does beta oxidation occur? Mitochondria. What part? Matrix. Perfect. So, the body must maintain a protein balance. So when we have excess protein, when we consume excess protein, that protein is broken down for other sources. The amino group is removed, leaving that carbon skeleton that I mentioned earlier, which will yield an alpha keto acid. Typically, if it's 5 carbons long, that will be alpha ketoglutarate. And those carbon skeletons are used as intermediates of the pathways that we've talked about in this block. And when the excess ammonia or the excess amino group is converted to urea and excreted by our kidneys. All right, any questions about that? So, what is this process called?
Transamination.
One person?
Transamination.
Transamination, correct. So in transamination, we're transferring that amino group from one substrate to another. So we see this amino acid is transferring its amino group to alpha ketoglutarate to produce pyruvate, which we know is the final product of glycolysis, producing glutamate, which is another amino acid. So with that, taking that into consideration, what is one of the functions of transamination? Talked about. Correct. Yes, absolutely. What else? Other person. Okay. Oh, yes.
What they say on there?
He said that it breaks down, the transamination will remove that amino group, allowing for the product substrate to be put into one of the pathways that we talked about, either the TCA cycle or glycolysis or gluconeogenesis for energy production or glucose.
I don't know if it's a purpose of transamination, but as a result, you'll get the glutamate will lose its ammonia group, and then it'll go into the urea cycle.
That's perfect. Yeah, that's correct too. What I was trying to get at is, you guys, what you said is 100% correct. It's also used to make non-essential amino acids. So remember we talked about there's 9 essential. We have to consume those all the time, and this is a process. Transamination will make those other 11 amino acids, as it's doing here. So alanine is becoming glutamate. Does that make sense?
So is that just are you just saying that like we're showing alanine here, but it could be a lot of different
Yes,
ones?
it could be all of the other 11 can undergo transamination
Okay.
to make another non-essential amino acid. So, transamination, you guys were correct. So, what is this process called?
Oxidative deamination.
Beautiful. Oxidative deamination. So, we're removing that amino group. It's becoming ammonia. And then where is this ammonia going to go? The urea cycle, correct. So, transamination, as you were alluding to, can be coupled with this step to remove excess ammonia. Does that make sense? All right. What about this reaction? It might be on your slide. I might not have taken it off. This is fatty acid activation. So in order for beta oxidation to occur, this step is the preliminary step that must occur that serves as the catalyst for beta oxidation. So a CoA group will attach to that fatty acid, producing that fatty acyl CoA group, which will then be transported via the carnitine shuttle into the matrix. Yes.
When that Cat 1 is not catalyzed this reaction, does it lose the CoA? Get another CoA?
Say again.
When that carnitine or the Cat 1 enzyme catalyzes the reaction?
So Cat1, before you finish your question, what enzyme catalyzes this reaction? It's acyl CoA synthase, not Cat.
I'm talking about like
Oh.
once it's fatty acyl CoA,
Once it's
does it lose that CoA once that Cat 1 catalyzes?
so the CoA will be, so there's CoAs in the cytosol and in the matrix. So it will lose that CoA, but then it'll get another one once it enters the matrix. Does that answer? So this enzyme that catalyzes this fatty acid activation is acyl CoA synthetase. So what that's doing is that this reaction is serving as the driving force for first this fatty acid, this activated fatty acid to be transported into the matrix by the carnitine acetyltransferase shuttle system and then beta oxidation. Does that make sense? Yes.
Sir, do we need to recognize the structure?
Sorry, thank you. I meant to say that. Absolutely not. No. Just know what's going on in the pathways and the enzymes. All right. What about this process? What is this called? Anybody? The urea cycle. So, of importance, the urea cycle, the goal is to remove that ammonium from excess amino acid intake to be excreted as urea, which will then be excreted in our urine by the kidney. So the urea cycle occurs in the liver. The first two steps occur in the mitochondria. That first step, the carbamoyl phosphate synthetase 1, is the rate limiting reaction of the urea cycle. So that product, carbamoyl phosphate, with ornithine and the enzyme ornithine transcarbamylase will produce citrulline. Citrulline will then enter the cytosol of liver cells to undergo metabolism into arginosuccinate, then arginine, then urea. So where did we see fumarate? TCA cycle. Yeah. So the important components of the urea cycle in terms of for what I want you guys to focus on is just the substrates of the urea cycle and also the locations that they occur in the rate limiting steps. Now, if we have a deficiency in the ornithine transcarbamylase, we will get conditions. We will have a symptomatology that includes hypotonia, that excess ammonium, hyperammonemia will enter the brain, mess with our motor system, so we will get positive asterixis, which means that we can't properly move our hands. There's motor functioning issues. We also see patients that get cerebral edema, because what happens in the brain is that ammonia, in efforts to try to break it down, it's done in the astrocytes. And what happens is astrocytes will convert that ammonia to glutamine. Glutamine is osmotic amino acid, so it will draw fluid into the astrocytes, causing cerebral edema, which we know have a number of negative side effects for So we would treat this condition by two drugs that basically act as ammonia scavengers. So they will inhibit the absorption of ammonia from our GI tract. And so that's helping to decrease the amount of ammonia that's in our body that can then go on to our brain since the urea cycle is compromised. And those two drugs are lactulose and rifaximin. Okay. Any questions about the urea cycle? All right. In terms of amino acid use for energy sources, those carbon skeletons, again, those carbon skeletons are the skeletons of amino acids without that alpha amino group, will be used as intermediates of glycolysis, ketogenesis, or the TCA cycle. So in general, you have 3 carbon amino acid. Their carbon skeletons will be used to produce pyruvate. You have a 5 carbon amino acid. Its carbon skeleton will be used to synthesize alpha ketoglutarate. And if you have a 4 carbon skeleton from amino acid, that will be used as oxaloacetate. All right. And so if these carbon skeletons are used to produce alpha ketoglutarate, acetyl-CoA, pyruvate, or any intermediate of glycolysis or the TCA cycle, they are considered to be glucogenic. If they lead to production of acetyl-CoA, those carbon skeletons, or acetyl-acetyl-CoA, which is ketone body, those carbon skeletons are said to be ketogenic. Any questions about that? Yes.
So then certainly only 6 ketogenic here that are in the yellowish tone.
Yeah, so the glucogenic ones are in salmon colored, the ketogenic are, and you don't need to focus on the names of them, just what they are producing. So and there is, if you look real closely, you don't, but if you look real closely, there's some that are found on both. So just in general, 3 carbons will produce pyruvate, 4 oxaloacetate, and 5 alpha. So those would be considered glucogenic. Any other questions about that? Yes.
Do we need to memorize the skeletons of each of these
No.
to be able to identify where they are?
No. In general, you have a 3 carbon skeleton that produces pyruvate.
What example? Like if I symbolize it, I don't know.
So since you don't have to know the structure, so I wouldn't ask for a specific amino acid. Does that answer it? Any other questions? All right. One of the stars of biochemistry, as I mentioned, phenylketonuria. We know that is a condition that occurs because phenylalanine hydroxylase is deficient. That is the enzyme that converts phenylalanine, which is an essential amino acid, to tyrosine. So because that enzyme is deficient, a transamination reaction occurs. So this is tying it together. This is connecting it more from the first block we just talked about products, but now we're seeing how transamination reaction is leading to those toxic products in the brain, and that is phenylpyruvate and phenylacetate. Those are toxic. So what you would want to do is if you had an infant that had PKU, you would ensure that you give them a diet low in phenylalanine, higher in tyrosine, so they bypass that step. That's one of the treatments. Any questions about that? Now, this is very important, cell signaling. So cell signaling, you'll see when we move to block 4 and talk about pharmacology, these pathways are targets of a number of pharmacotherapeutic interventions. So that's why they're so important. And so we talked about an intracellular cell signaling and surface cell signaling. So we know that intracellular cell signaling occurs via steroid hormones because they're lipophilic, so they can enter the cell membrane of these. They either have an intracellular receptor in the cytoplasm or even in the nucleus. And what happens when they bind to whatever their receptor system is, that's going to cause some type of gene regulation, either upregulation or downregulation of a particular protein. And so that will be the biological response from intracellular signaling pathway. Now for the cell surface signaling, we know that ligand or molecule will bind on the cell surface to a receptor system bound on the cell surface, initiating cascade of events through a second messenger system. So we talked about cyclic AMP, calcium, and diacylglycerol as second messenger systems. We're going to talk about this one in block 4. But these second messenger systems, depending on what receptor system this is, will either upregulate or increase cyclic AMP, calcium, or diacylglycerol, leading to protein modifications, enzyme modifications, and a biological response. Any questions about that? So we talked about the G protein couple receptors. So the G protein couple receptors would be the surface receptors. And so we talked about 3 different types of G couple protein receptors, the G sub S, G sub I, G sub Q, and how they are coupled with receptor systems. So, for example, G sub I is coupled with these receptor systems, MAD2, that's muscarinic 2, alpha adrenergic 2, and dopamine 2, and their ligand or agonist that binds to the extracellular protein found in parentheses. And so once that reaction occurs, it will inhibit cyclic AMP production and then produce the respective physiological responses. So this is very important, knowing what protein systems are coupled to G sub S, G sub I, and G sub Q. All right, so if we look at G sub S and G sub I in particular, we know this mechanism. What's happening is so if this was the dopamine 1 receptor, dopamine would bind to the receptor, initiating the dissociation of the alpha subunit, and the beta gamma dimer would form. That alpha subunit would then activate adenocyclase, which will cause the conversion of ATP to cyclic AMP, and then that will lead to the respective physiological effects of D1. Conversely, we have an alpha 2, which is inhibitory. Norepinephrine or epinephrine will bind, causing that same dissociation of the alpha and the beta gamma dimer. But in this case, that alpha subunit will inhibit adenocyclase. So cyclic AMP will not be produced, which will lead to a set of physiological responses. So remember, when looking at these two, G sub S and G sub I, they're producing opposing effects on the cell. Does that make sense? All right. You guys have any other questions? Yes.
Sir, you still need to know the process for the G sub substrate Q?
Yeah. Yes. Yeah. Do you know it? Can you say it?
Once it binds, the DAG splits off from the IP.
Yeah, that's pretty close. I'm gonna pull it up. But yeah, great job. Let me see. All right. So in this case, if you had vasopressin would be the receptor, antidiuretic hormone would bind, causing the dissociation of that alpha subunit from the beta gamma. Phosphatidylinositol bisphosphate will be cleaved into DAG, which will hang out in the cell membrane. That IP3 will then enter the cytosol to cause calcium release from that endoplasmic reticulum and subsequent physiological effects. Also, that DAG will activate protein kinase C to cause a set of physiological effects. So, great job. Any other questions?
Do we need to worry at all that GI is mentioned to also simulate phospholipase C?
No.
Don't worry about that part.
Yes.
So, sir, the alpha, beta, and gamma subunits, they kind of always are bound to GDP, correct?
Yes.
And then when it's converted to GTP is when the alpha subunit breaks off.
Yeah. Great question. Thank you. Any other questions? All right. If you guys have any questions, email me. I'll be on standby this weekend. If you have any burning questions for me. Or you can come to my office, first floor, room 1319. All right. Good luck on your test on Friday, guys.